Welcome to topic 3, virus quantification. This content is part of the module MBS 7117, Molecular Microbiotechnology. It's housed in the Department of Biomolecular Lab Technology and Molecular Biology, abbreviated as BLT, in the School of Biosecurity, Biotechnology and Laboratory Sciences, SBLS, in the College of Veterinary Medicine, Animal Resources and Biosecurity, COVAB, in Makerere University. I am your presenter today, Dr. Penny Nansamba. Our content for topic 3 consists of the following. Number 1, definition and justification for virus quantifications. Number 2, types of assays for the quantification of viruses. Number 3, plague titration assays. And number 4, infectious dose 50. We are now in the third series of topic 3, and we shall be concentrating on the content for infectious dose 50. The learning objectives for topic 3 are as follows. By the end of this lesson, you'll be able to, number 1, justify virus quantification. Number 2, list methods for virus quantification. Number 3, explain the plaque titration. And number 4, explain the infectious dose 50. This being the third series in topic 3, we are going to concentrate on learning objective 4, explaining the infectious dose 50. So, series 3 in topic 3, infectious dose 50. Learning objective 4. By the end of this section, you'll be able to explain the infectious dose 50, highlighting the principle underlying it. Infectious dose 50. Let's start with the definition and its application. So, number 1, infectious dose 50 is a quantitative measure. Secondly, the dose of a virus required to infect 50% of a group of exposed hosts. It may be animals, it may be eggs, or it may be cell culture. Infectious dose 50 does not measure the total number of virus particles, but rather the number required to cause an infection in 50% of the test units. Lastly, infectious dose 50 is commonly used in animal studies, vaccine testing, and assessing pathogen virulence. There are many types of infectious dose 50. We are going to look at these. TCID 50. This is the infectious dose for 50% of the tissue culture cells to become infected. EID 50 is the infectious dose for 50% of the embryonated eggs to become infected. LD 50 is the lethal dose causing 50%, LD 50 is the lethal dose causing death in 50% of the experimental animals. ID 50. This is the dose infecting 50% of the experimental animals. Now let us illustrate the procedure for TCID 50, tissue culture infective dose, where 50% of the wells in a multi-plate will become infected or show CPE. So we're going to illustrate this procedure for you to understand this. So number one, we're going to do a serial dilution of our virus. We are going to do a serial dilution of our virus, tenfold dilutions, because that is easy to handle. So we get our tubes, dilute stock at 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, and so on up to 10 to the minus 8. We saw this in our previous series too, which you can refer to if you want to learn more about the serial virus dilutions in tenfold dilutions. So, step 2, we are going to inoculate a 96 well plate. The picture below shows us a pictorial of an artistic view of a 96 well plate. Remember, we have diluted our stock viruses in tenfold dilutions. So we're going to get our virus diluted at 10 to the 1 minus 1 and fill it in all the wells in column 1. We're going to get our virus diluted at 10 to the minus 2 and fill it in the column labeled 2. When you use a multi-channel pipette, it's quite easy and there's less room for error. We are going to get our virus diluted at 10 to the minus 3. Using a multi-channel pipette, use it to fill the column 10 to 3. And we are going to go on until, we've got to column number 8 or column number 7 and filled it with virus diluted at 10 to the minus 7 or 10 to the minus 8. After this, we add viable cells onto our 96 well plate. Then we go to step number 3. We are going to incubate our 96 well plates in an incubator, in a carbon dioxide incubator. It will be at a temperature of 35 degrees Celsius to 37 degrees Celsius. The carbon dioxide concentration should be 5%. We incubate these plates for about 1 week and observe for CPE. Step 4, we are going to observe our plates for CPE. There will be some parts where there is visible CPE, so there are no cells in these wells. They've been indicated in color yellow here. Then there will be some wells where there was no CPE, and these will still have cells, and we can stain them with methyl blue, with gentian violet, and they will appear purple because the cells will take up the color of the dye, purple. So let's interpret these results and calculate this TCID 50. The endpoint where 50% of the wells have been affected by CPE and 50% of the wells have not been affected by CPE. So we are going to record the positive wells that have been affected by CPE at each dilution. 10 to the minus 1, 10 to the minus 2, and so on up to 10 to the minus 8. We are also going to record the number of wells where no CPE took place, and these are negative wells at each dilution also. 10 to the minus 1, minus 2, minus 3, up to 10 to the minus 8. Now we can use a statistical method like the Reed-Mureck or Spearman's-Caber to calculate the TCID 50 for us, or the endpoint at which 50% of the wells were infected. But we can also look at a manual way of doing it. It's not so accurate, but it gives you a better insight into the TCID 50. So number 1, we are going to record all the wells where we had CPE, taking note of also all the wells that we had no CPE. And we are going to record this graphically. So our endpoint would be around between It can't be 10 to the 1 because we see all those words have got CPE. It can't be 10 to the 9 or 10 to the 10 because we see all those words are purple and they don't have CPE. It would be in the middle, maybe around 10 to the 6 or 10 to the 7. But let's calculate this manually using a graph. So we have our graph, and we're going to take our first dilution, 10 to the 1. All of those words had CPE, and on our graph, we're going to record this percentage of words with CPE at 100%. The log dilution is 1. We go to 10 to the 2. All those words had CPE. On our graph at the log dilution of 2, we show 100% CPE. We go to 10 to the 3. Out of the 8 wells, 7 had CPE. So what percentage is that? 7 out of 8, that's about 88%. And 10 to the 3, we shall record that, about 88%. We go to 10 to the 4. It's the same, about 88%, because we only see one well that didn't have CPE, while the other 7 wells had CPE. We get to 10 to the 5. 2 wells do not have CPE. That means 2 out of 8. 6 wells show CPE. So what is the percentage of the wells with CPE? 6 divided by 8, that's about 75%. So we are going to record that at about 75%. 10 to the 6. More wells are showing no CPE and less wells are showing CPE. So 3 out of the 8 wells do not have CPE. That means 5 out of the 8 wells show CPE. So 5 out of 8 showing CPE, that's about 63%. We have recorded that. 10 to the 7. Now we have 3 wells not showing CPE. That means 5 wells show CPE. We have at 10 to the 7, 3 wells do not 3 wells show CPE. I beg your pardon. At 10 to the 7, 3 wells show CPE and 5 wells do not show CPE. So the percentage of the wells showing CPE are about 38%. Our graph is going lower. At 10 to the 8, now 2 wells show CPE. 2 out of the 8 wells show CPE. That's about 25%. 10 to the 9, only 1 out of the 8 wells shows CPE. So that's about 1 divided by 8. That's about 12%. When you get to 10 to the minus 10, none of the wells show CPE, so that is 0. The column 11 and column 12 were used as controls. They only had cells with no viruses. So how are we going to get the TCID 50? Using this graph, we want to get to the point where 50% of the wells showed CPE and 50% did not show CPE. So using the x-axis, we are going to look for the 50% mark and draw a line towards the graph. Then draw it, extrapolate it to the x-axis, and we shall see the log dilution at 50% is between 6 and 7, which is about 6.5. That is where we had originally estimated it to be. So that's the TCID simply. 50% of the wells showing CPE and 50% of the wells not showing CPE. But usually we use statistical methods and we record the positive wells with no dilution. I mean positive wells with CPE at each dilution, the wells with no CPE or negative wells at each dilution, and feed this into a formula, and it gives us the endpoint, TCID 50 endpoint. So let's look at the strength and weaknesses of the TCID 50. Advantages: the viruses that do not form plaques or that do not infect cell monolayers can be quantified, which is good. It can be used when virus-specific antibodies are not available for many ELISAs. There's a minimum error compared to the plaque assays, and there's a high degree of sensitivity and accuracy. However, for each time we have an advantage, there are also disadvantages. It is lengthy, time-consuming, taking several days, and we need technical skill to read and calculate the results. So the next activity is I want you to visualize the procedure for conducting a TCID 50 count using this video, and it's by the Virology Research Services. The URL has been shared with you. It's going to take about 4.5 minutes. So briefly, let us reflect on what we've just learned. Our question today is compare and contrast the plaque assay to the TCID 50 assay in terms of methodology, sensitivity, result interpretation, and its applicability. As a hint, you're going to look at the ease and the costs. We are secondly going to look at the type of virus for which this method can work for. Thirdly, we are going to look at the turnover time, the time it's going to take for us to get the results. Fourthly, we're going to consider sensitivity and reproducibility for each method. We are going to look at the output units that we want. Are we going to look, do we want to do plaque forming units, or do we want to do the TCID 50 in mils per liter? And then we can look at whether we want to assess infectivity, which is seen in the plaque assays, or whether we want a statistical estimation, which is seen in the TCID 50. Now, this will be a take-home exercise. This is quite lengthy. So in summary, the lessons learned is the procedure for carrying out the TCID 50 and also its merits and demerits. The next steps: refer to the lecture notes given to you on your study portal, the activities therein, the chapters in the textbooks that we have recommended to you for further reading, and any other peer-reviewed journals that we have recommended to you for further reading. These are our sources of information. Thank you.